Okay, we're going to continue with our digestive system notes, and we're going to start with the pancreas. Now, the pancreas is a gland that is considered both endocrine and exocrine in nature. Endocrine means that it releases its products within the body itself. The way that it does that is some of the products that the pancreas are going to release are going to go into the bloodstream, and that's what makes it endocrine in nature. It's exocrine in nature because some of the products that the pancreas has to release are going to get released into the lumen of the small intestine, which even though it's not technically outside of the body, because our alimentary canal is truly exposed to the outside of the body from mouth to anus, it's kind of like being secreted outside of the body. I know that's kind of weird to think about it, but if you imagine it, really it's almost like our body is around a tube. Anyway, the pancreas is nuzzled into the duodenum like this, and it's kind of got this little tadpole looking structure to it, and in fact this part is called the tail, and the rest of it's the body. And if we were to look at the abdomen with all of our abdominal organs intact, the stomach would be right there in front of it. Now, the histology of the pancreas, remember, histology is the study of tissues, and in the pancreas we have functional units called islets. These are called islets of Langerhans, and they are also called pancreatic islets because we always have to have the name of the scientist and the name that science uses. So the large islets are where we find the endocrine portions of the pancreas, and those are cells that we might be familiar with because of people that we know that might have diabetes. There's one type of cell called alpha cells that produce a hormone called glucagon, and glucagon's structure is to increase glucose blood levels when they're low. There's another type of cell inside the islets called beta cells, and beta cells produce a hormone called insulin, and insulin decreases blood glucose levels when blood glucose levels are too high. This is a feedback mechanism that works with the liver in order to maintain proper blood glucose levels. Remember, we always want homeostasis, we always want things to be in balance. The rest of the pancreas are made of acini, which are the majority of the structure of the pancreas. 99% of it is actually what we call exocrine in nature, and it contributes to pancreatic juice. Pancreatic juice is going to be that digestive part of the pancreas that's going to contribute to helping to continue to break down our food. So what's in pancreatic juice? Well, we secrete a lot of it every day. 1.5 quarts a day, and the pH is roughly around 7.1 to 8.2, which means we are in the slightly basic range. And it's got water, but the big thing it's got is our enzymes and sodium bicarb. Sodium bicarbonate is basic because when we take the acid that we just had and we add a base to it, we get to neutral. So we need to neutralize that acid that's coming out of the stomach real quick, because if we do not, we will have that acid starting to burn a hole into the duodenum. So what kinds of enzymes are in pancreatic juice? Well, there's pancreatic amylase, which digests carbs, pancreatic lipase, which digests fats, and then several enzymes that are that will contribute to digesting proteins. In the stomach, we just had the one called pepsin. But from the pancreatic juice, we have trypsin, chymotrypsin, and carboxypeptidase. Lots of fun names there. Just like the pepsin in the stomach, these proteins are released in their inactive form, and they become activated by another enzyme called enterokinase, which is just Latin for get it moving inside. Entero means inside and kine means to move. Anyway, these are released in their inactive forms, and this enzyme activates them again because we don't want the protein digesting enzymes to actually start digesting our own body. And then, this is the first time we see anything that contributes to digesting our nucleic acids. Our nucleic acids are DNA and RNA. If you remember from Baby Bio, these are our genetic material, so anytime you eat something, you're going to eat its DNA. So what we were just looking at it with the pancreatic juice was the exocrine portion of the pancreas's job. The endocrine portion is all about blood sugar levels. And we need sugar in our blood. It's really important for our brain. But what's really important is that those levels are regulated. Too much is very bad. Too little is also very bad. So this feedback system, this example here is going to start if we have low blood glucose levels. Low blood glucose levels is known as hypoglycemia. So if our blood levels are low, then the cells that are in the pancreas, the alpha cells, are going to release a substance called glucagon. Glucagon travels down to the liver, so this was the pancreas. There's my wonderful drawing of the pancreas. And it's going to act on the liver. It kind of looks the opposite of the pancreas. And the glucagon goes into the liver and tells the liver to break down glycogen, which was a storage product for glucose in animals, into its subcomponent called glucose. There's also other types of stored energy. There's lactic acid, there's amino acids, but the one that our body kind of uses the most is glucose. So the liver releases the glucose into the blood, and that's going to allow blood glucose levels to go up. So if it goes too high, of course, we have to inhibit that action. High blood sugar levels are called hyperglycemia. And when that happens, we get a different type of cell. We get insulin, which is released by the beta cells. What does it do? Well, it doesn't just stop the glucose from being released, although that is one of the things that happens. First, it allows the cells to actually grab more glucose out of the bloodstream so that it can get out from floating around in the blood, and particularly it'll go into skeletal muscles. It will also tell the liver to start reconverting glucose into glycogen and store it. It will also increase amino acid production by cells, and that in turn increases protein synthesis. Amino acids are the building blocks of proteins, and this process right here of building the proteins takes energy, and in order to get that energy in the cell, you got to have glucose. So all of those things will help take some of that sugar that was in the blood and get rid of it. As a result, blood glucose goes down, and if we fall too low, we start the cycle over again. Now, this is a really important feedback mechanism, because any of you guys who know somebody who has diabetes, this system is not working properly. And we're going to talk more about that in class. But what's important here is how the cells regulate themselves and which cells are doing what. This is an essay question. Yay! So how the heck are you going to remember all this? Well, you can memorize this fancy chart. It is quite beautiful. Or you can remember this. Lag Hibby. Low blood glucose, alpha cells, release glucagon. Hibby, high blood glucose, beta cells release insulin. So, of course, that doesn't explain the whole story. Perhaps the trickiest part is knowing the difference between these terms. Glycogen, glucagon, glucose. Glycogen is our storage of carbohydrates. Glucagon is the hormone that's released from the pancreas when we detect low blood sugar levels. And glucose is the building block of all carbohydrates in the body, or one of the building blocks of carbohydrates in the body. And this is what is going to be released when glucagon is released into the bloodstream. So this is the storage. This releases the glucose, and then the glucagon releases the glucose from storage. Now, granted, there are more steps involved in this, but this is at least one way to remember what's going to react in the situation of low or high blood glucose levels. Okay, so let's talk a little bit about the liver. Liver, anatomically speaking, is one of our largest organs. Some of you guys were surprised when we did the rat dissection by how large the liver was. In our body, it weighs about 3 pounds, and we have 2 lobes, and the right lobe is larger, and it's separated by a ligament called the falciform ligament. Then we have the gallbladder, which is posterior to the liver, and it is a little pear-shaped sac in the liver that concentrates and stores bile. And you will always see it colored green in any diagram you ever see of the liver and gallbladder. Histologically speaking, the liver, we're not going to spend a whole lot of time on the histology of the liver, but we should know what the name of liver cells are. Liver cells are called hepatocytes, and their function is to secrete bile. There's little tiny ducts called canaliculi. We saw that word before when we were studying the skeletal system. And this is where the bile that the hepatocytes produce are going to start collecting in there, and then eventually it'll make its way to the gallbladder for storage. There are little sinusoids, which are capillaries that are between all the hepatocytes that feed the liver cells. And then there's Kupffer cells, and of course this is the guy who discovered them, and here's the actual science name. Stellate reticuloendothelial cells. I think Kupffer might be easier in that case. Stellate because it's star-shaped. And these have a really important job because their job is to get rid of blood cells that are all worn out, destroy matter that's gotten into the body, toxins and microbes, and they're part of the detoxification part of the liver. Okay, so what is bile? Bile is a liquid that's made up of water, bile salts, cholesterol, lecithin, and some pigments. And its really important job is emulsification. And you guys were asking me that earlier. And what that basically means is that it's going to contribute to breaking down large fat molecules. Fats can be very, very big molecules into smaller pieces so that they can be absorbed into the intestine. It's not exactly the same as digestion per se, but it's really important because it increases the surface area of fat and allows it to become available to be digested by enzymes called lipases, which will break down the fat further because they cannot get to the lipid droplets in the form that they're in until they're emulsified first. So it's a really important job that they do. The pigment that's in bile is called bilirubin, and bilirubin comes from the heme group, which is a group that is a part of the red blood cells. And this is the pigment that will eventually cause your feces to have a brownish color because it starts as bilirubin and then becomes stercobilin, and that'll kind of make your poop look brown. So lots of functions in the liver. Make sure that you pause it to get all of the notes that you need for the liver functions. We already talked about it maintaining blood glucose levels. It also contributes to the metabolism of lipids, not by digesting them, like I said, but breaking them down so that they can eventually be used for energy. They also help metabolize proteins. We didn't talk about that much at all, but the liver itself helps remove NH2, which is it's called an amine group, from amino acids. And what we get left is energy. It converts ammonia into urea. And it also makes some of the proteins that we use in our clotting mechanism. The liver, you've heard of the liver as a detoxifying center, so it helps. That is the place where any drugs, be it legal or illegal, are taken, or over the counter or not, are taken to the liver to remove any products that are going to be toxic to you. It's one of the reasons why livers will fail over time by people who abuse drugs and alcohol or other over-the-counter medications. And that includes hormones as well. So if you take a birth control pill or if you're menopausal and you're on other types of hormone, it's going to be the liver that's going to take care of that. Get rid of bilirubin, store some minerals, particularly what we call our fat-soluble vitamins, which is A-DEC and B12, and then storing some of our minerals, iron and copper in particular, and then activation of vitamin D. The skin takes care of this as well, but when you're not exposed to sunlight, then the liver will do that as well.